Moving on to section 2 of your video notes over DNA replication. Before we dive into DNA replication, I wanted to talk a little bit about the cell cycle. This image here depicts the life cycle of a cell and different important things that happen along the way. You can imagine that the birth of a cell happens right here in G1. The cell spends most of its life in this first stage we call interphase. Interphase is broken down into 3 separate parts. So when a cell first divides or is born, we would say it begins to grow. It grows and it makes organelles during G1. Now, once the cell gets large enough, it's getting ready to start the process of dividing or becoming 2 new daughter cells. At the end of G1 or gap 1, there is S phase. We also call this the synthesis phase. This is where the cell continues to grow, but something very important happens in that DNA replicates or makes a copy of itself. It is important to point out that whenever cells divide, they all need a complete set of DNA or chromosomes. Following the S phase is the third phase, G2 or gap 2. In this phase, the cell continues to grow and makes final preparations to begin to divide. So we'll talk about mitosis in its own section, which is these stages right here. This is the part where the nucleus of the cell divides. Lastly, in cytokinesis, just a quick preview, is whenever the rest of the cell divides, so the cytoplasm, the organelles, and the membranes break off from each other to form 2 new daughter cells, and then the cycle again repeats. So in this slide, just reviewing the 3 parts of interphase, this is where the cell spends most of its life, and it's broken down into these 3 parts as you see here. The first gap phase, the synthesis phase, and the second gap phase. The first gap is when the cell grows and it makes copies of organelles. The synthesis phase is when DNA replicates, which is an important process that we'll talk about in this section of notes. And then finally, after replication is complete, the cell continues to grow and prepares for division in the second gap phase. So let's quickly overview DNA replication. You'll need to remember the different structures of DNA that we talked about in the first section of video notes. But let's kind of break down some major parts. First, DNA replication, as you guys know, occurs during the synthesis phase of interphase in the cell cycle. It occurs within the nucleus of the cell as that is where DNA is kept safe. We say this is a semi-conservative process. What that means, as you can see in this image right here, you've got 2 DNA strands or a double helix. These are your original strands of DNA. Then they break apart and they are used as a template to make copies off of. So breaking the bonds between the bases to use these blue strands as a template for the new daughter strands of DNA. That is why we say it is semi-conservative, and that whenever the DNA is completely done making copies, each strand of the double helix includes the original parent strand, and then the other strand is the new daughter strand. It is important to point out that in the cell cycle, there are many checkpoints along the way to prevent copying errors, and we'll talk more about those. Those are called mutations. So the first step of DNA replication is strand separation. So before you can make a copy of DNA, you've got to break these bonds between your nitrogen bases. And remember, these are hydrogen bonds between the bases. So cytosine and guanine, each of them have 3 hydrogen bonds, and adenine and thymine each have 2 hydrogen bonds between them. Helicase is an enzyme, remember, a protein with a specific function, and it ends with ase, so that's how you know it's an enzyme. It breaks these hydrogen bonds between the 2 parent strands of DNA almost like a zipper would work. The point where these 2 strands separate from each other is called the replication fork. So you can see at the replication fork, these bases are exposed so that new nucleotides can be added to complement them. That leads us to the second part of DNA replication, base pairing. So DNA polymerase is another enzyme that's really important to this process. And in the nucleus, there are these free-floating nucleotides. So these nucleotides for DNA that include the 5-carbon sugar, the base, and the phosphate group. They get added by this enzyme to complement the exposed parent strands of DNA. So the daughter strand will be added 1 base at a time by the DNA polymerase. So here a thymine would be added, here a guanine would be added, here an adenine would be added, and so on and so forth. And the same process would occur on this parent strand, where and adenine would be added, cytosine, then thymine, and adenine, and guanine. So that's how this process works. And again, DNA polymerase is your proofreading enzyme. So it's also going to check for errors while it's adding nucleotides. This is a very energy-intensive process, so it does require ATP to make the copies of DNA. Remember, the cellular form of energy is always going to be ATP. Step 3 of DNA replication is whenever everything gets kind of sealed together. So a different enzyme is used here called ligase. Ligase will seal the bonds of the newly formed DNA together, so it strengthens the bonds between nucleotides and the bases. This process results in 2 identical copies of DNA, almost like a complete set. Each new DNA molecule, again, is composed of one of the parent strands and one of the daughter strands. This is why we call it a semi-conservative process.